Hello, everyone, and welcome to today's webinar, Cell-Based Assays to Functionally Characterize Immunotherapy and the Immune Response to Viral Infection. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Chris Langsdorf, staff scientist at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Chris, you may now begin your presentation.
Great, thank you for the introduction, and indeed thanks to LabRoots for hosting this webinar. So it looks like we have quite a few people on the line, and thanks to you all for joining us. I realize these are very busy times as we're getting back into the lab and resuming our research. I know in our lab it's very busy as we're continuing to pick up new projects, and so today we'll discuss a lot of our research, specifically around two topics related to immunology. First, immunotherapy, and second, how we can study the immune response to viral infection, and especially looking at how we can use cell-based assays to study how viruses infect cells and replicate. Now, before we begin, I want to announce the launch of a new website that came out recently called the Immunology at Work Resource Center. This is a free resource designed for scientists of all levels to provide an overview of immunology in all its aspects, foundational immunology, different immune cell subsets, viral immunity, along with protocols covering a lot of what I'll discuss today. So this is meant for a learning resource for you, realizing that a lot of our work has pivoted towards immunology in some aspect. So this provides a good overview of the field. Now for today, and actually this figure is drawn from the Immunology at Work Resource Center, where in immunity, we have a variety of different cell types that protect us, either from cancerous cells or from pathogenic infection. We can break these down into both innate and adaptive cells and functions. And today we'll discuss cells from both of these compartments. Specifically, we'll focus on natural killer cells and on T cells. And start out by looking at how these two types of lymphocytes are able to function either in their natural setting to target cancer cells, as we see down below, through MHC complexes and recognition of presented peptides, or up above, where we can reprogram these cells either with chimeric antigen receptor T cells or using therapeutic antibodies against cancer cells to encourage natural killer cells to specifically target and lyse cancer cells. And so we'll go over a range of different protocols for not only isolating these different cell types, expanding their number in culture so you have more to work with, and then matching up these immune cells against various cancers and show how we can study their behavior and how we can modify that behavior using different therapeutics. So let's start off by looking at T cells. Now, T cells, of course, are easily obtained either from mouse or human whole blood. Very often these days, we're working more with human whole blood. So often what we'd prefer to do is from whole blood, we'll use a density gradient to separate out the peripheral blood mononuclear cells, as you see in the white layer here in this centrifuge tube. After we isolate those PBMCs, we can use a couple of different approaches to then isolate our T cells. Shown here on the right are Dynabeads untouched human T cells. Now, these contain essentially tiny magnetic beads and antibodies against every blood cell type except for T cells. So essentially, we will deplete all of the cells in our blood except for T cells. This becomes important if we would like T cells that are not activated but are isolated. If we plan to activate our T cells anyway, we can use something different. This is called the Gipco CTS Dynabeads CD3 CD28. Now, these are the same magnetic beads, except they're now coated with antibodies against human CD3 and CD28. So this will do two things. First, it will positively isolate T cells from your PBMCs based on the CD3 binding to this T cell receptor. Second, after isolating the T cells, this will very aggressively activate and expand, meaning encourage our T cells to proliferate by means of CD3 antibody providing a stimulatory signal and CD28 providing the co-stimulatory signal, encouraging a robust proliferation of our T cells. And so let me show you what this actually looks like in a culture dish. If we were to take PBMCs and use these CD3 and 28 Dynabeads to isolate T cells, and then just plate them here in a T75 flask, we can see the beads are not that much smaller than a T cell. We can easily visualize them both here at 40x on our XL Core microscope. Now, immediately after plating, both of these components, the beads and the cells, will settle to the bottom fairly quickly, and we can image them. Now, if we were to put these in an incubator overnight and not agitate them, but just let them sit, we come back the next day and we can see these clusters have formed. And this is due to beads binding to a cell and also binding to a neighboring cell, and that cell binding to additional beads. And essentially we see these clusters begin to form. If we come back and look the following several days, we see that these clusters substantially increase in size. And I find this valuable because you can actually see these without even a microscope being required. Just by eye, you can see these aggregates form, and it tells you that it's working. It tells you that our T cells are effectively proliferating. And we see this with natural killer cells also, or with even CAR T cells. We expect to see the same thing. Now, this is not a spheroid or an organoid by any means. It's just a loose association of cells. And so we can either swirl the media in this plate, or we can use a very large pipette, like a 25 mil pipette, and just aspirate up and down the cells and the media, and it will disrupt these structures back to single cell suspension. Then we can put these in a conical tube in a magnet and pull the beads out. And what we're left with is purified and activated and expanded human T cells that we can take and put into our downstream experiments. Now, at this point, in this example, these cells have been out of a human now for 5 days. And so we'd like to do a quick quality check to see how healthy are they, have they died, or are they viable, and is it a good idea to carry these on into our downstream assays. And so I'd like to announce a new instrument that will be launched next week called the Invitrogen Countess 3 Automated Cell Counter. So this is the third generation of a cell counting device where you can take 10 microliters of your cell sample, insert it into a small plastic chip that you see in the middle frame there, and within about 10 seconds, you can get a measurement of your cell concentration and of percent live, percent dead, or if they're labeled with any fluorescent dye, it can report on the fluorescence intensity of single cells as well. This incorporates a lot of new features, better optics, better software, machine learning to help identify what is a cell and what's not a cell to give you more accurate results. And so this is something that I use, many times throughout the day in the lab at every step to measure both concentration and viability of our cells. Concentration measurement is useful to report, to better understand how much our cells have proliferated, what the fold expansion is. But there are other ways we can measure that expansion or proliferation of our cells. One very common method uses a technique called dye dilution, in which we will label cells, and often this is used with T cells. We'll label cells with a very bright dye, which represents the brightest, farthest right peak on this schematic of a fluorescence histogram. Now, every time those T cells divide, half of the dye is passed to each daughter cell, and so we see an additional peak on our histogram that is half as bright as the parent peak. And so if our T cells were labeled with a dye such as cell trace violet and either left unstimulated or stimulated, in this case, with an antibody against CD3 and with interleukin-2, we can see the appearance of, in this case, 10 different generations of cells. So we can still visualize our parent generation, and that unstimulated control is valuable here so that we can see where the parent generation is, but now we can actually characterize the proliferation of our cells. And I've included a QR code here. If you hold your phone in front of it, it will take you directly to a good protocol for using cell trace violet to study expanded T cells. Increasingly, we're finding use for these cell trace dyes in other applications. And one of those is when we want to take immune cells, such as T cells or natural killer cells, and combine them with cancer cells that we'd like to study how they're being killed. And what we find is, in the case of mixing T cells with, for example, a leukemia sample, they're very similar in size and shape. And so they're difficult to identify which cell is which. However, if we were to pre-label both our T cells and, in this case, our target leukemia cells with different colors of cell tracing dyes, and then mix them together, we can now see that our T cells show up as blue and our cancer cells show up as red. And so now we have a very simple potency assay where we can add a third reagent called the cell event caspase 3.7 sensor. And now as our T cells kill our cancer cells, the cancer cells will turn green. So we can observe this experiment over time and quantify or visualize our T cells killing our target cancer cells. Now, often with this type of model, flow cytometry is preferred to provide a quantitative single-cell analysis of a lot of cells from a lot of different samples. And so we can use this exact same model with flow cytometry to provide a good high-throughput measurement of T cell killing. And so in this case, we're doing the same experiment where we're labeling some K562 cells with our cell trace violet dye, and we'll mix them with T cells and let them sit for 1 hour. And then we'll add a viability dye called Cytox Deep Red. This is a dye that shows up in your Cy5 channel, and it will only enter dead cells and make them show up very brightly red. And so in this model, we can very easily put a gate on our cell trace violet positive cells to identify our cancer cells. And then we can just ask, how dead are those cells? And in the histogram on the right, we can see after only 1 hour, almost half of those cells have been killed. Now, a couple of important things to note about this setup. One is that we've chosen 2 very bright dyes, and so it's easy to set gates on these and then apply those gates to, say, 96 different samples. And so that's where high-throughput analysis comes in. Also, we've done all of our cell labeling during the assay. So there's the endpoint, we just analyze. We're not doing any antibody labeling or washing or fixing or perming. As soon as our hour is over, we put our sample into our Attune Nxt flow cytometer and analyze immediately. And so we can capture accurately that endpoint and know that our time is correct. Now, in this example, we're looking at T cells killing one cell type, but we know increasingly in immunotherapy, chimeric antigen receptor T cells are used because they can specifically target a T cell to recognize cells expressing a given antigen. And often this antigen is CD19, which is expressed on both healthy B cells and a variety of B cell derived cancers. And so what's important here is that not only does the CAR T cell specifically kill a target cell, but it does not kill cells that lack that receptor. And so it's a good targeted therapy. To study this, we could expand the assay we've just described with an additional color so that we can use 3 colors of cell trace dyes to label each of these 3 populations of cells. Then mix those 3 cell types together, and in this case, we'll use a different viability dye, Cytox green, to indicate killing. Now, importantly here, a few things to note about this brief protocol. All of these cell trace dyes are recommended at somewhat higher concentrations, around 5 micromolar, for doing a proliferation analysis. In this example, we want to use a much lower concentration. I would use no more than 1 micromolar or even 500 nanomolar dye. Because these dyes are so bright, it becomes hard to get them on scale. So use a lower concentration of these. Use a normal concentration of Cytox green, which is in the high nanomolar also. Then we'll just let these sit together for 8 hours. And then after 8 hours in an incubator, we'll analyze and see what happened. So we'll take this 96-well plate directly out of the incubator, put it into our Attune Nxt autosampler, and see what results we get. Now, important to point out, we're using 3 different colors of cell trace dyes to label 3 different populations of cells. This gives us the ability to very easily distinguish cells from each other and very clearly. And so with flow cytometry, what we see is 3 different populations of cells. On the top left, we have our CD19 positive population. On the bottom right is our CD19 negative, what we could call the bystander population. Now, if we gate on our target cells and ask how live are they, or to what extent have they been killed, we can see that in this case, 32% of those cells are dead at the end of this experiment. By comparison, our CD19 negative cells have a significantly smaller fraction of dead cells, 9%. And so we can see in this example that we can easily characterize the viability and identity of different cell types in this T cell killing assay. All right, so we can move on now beyond T cells and look at, again, many in the field are pivoting from a focus on the adaptive immune system and T cells to the innate immune system and the innate immune response to cancer as exemplified by natural killer cells. Now, NK cells, of course, can also act through a variety of activating and inhibitory receptors to both recognize and kill either cancer cells or those cells infected by a virus. Or we can use a therapeutic antibody to encourage those NK cells to bind and kill. And so a closer look at that model, if our cancer cell expresses a given antigen and we have a therapeutic antibody that targets that antigen, importantly, this should be a human anti-human or a humanized anti-human antibody, so that the CD16 or Fc gamma receptor 3A on our natural killer cells will bind to that antibody and then specifically kill our cancer cells. And so I'll show you a bit of detail on how we study this interaction and this mechanism of immunotherapy. But first, to briefly mention how we acquire natural killer cells in the first place. And so much as we did with T cells, let's start with peripheral blood mononuclear cells. And in this case, we'll use Dynabeads untouched human NK cells. In this case, you will take your PBMCs at a very high concentration, label them with an antibody cocktail, and then after washing, you'll add functionalized Dynabeads, which will bind and pull down or deplete all of the other cells in your sample. I did this yesterday in our lab, and it takes about an hour to produce unmodified, untouched human natural killer cells. And so as you see in this picture, if you choose to expand your natural killer cells, they will form these cluster structures, much as you see with T cells. So morphologically, under a microscope at 40x, they look quite similar to T cells. However, they do express a variety of different receptors. And so we may prefer, in this case, we did expand our natural killer cells for 15 days. And we'd like to do a check of their viability and their phenotype before studying their function. And so with flow cytometry, this can easily be done with, in this case, a dye called Cytox Blue. And we can see that our natural killer cells are doing very well. They're 98% live. And then as far as phenotype, we can see in this cell population, the vast majority express CD56 and do not express CD3. And so we can say this is quite a pure and live population of human natural killer cells. And so based on this, we feel comfortable carrying these cells forward into a characterization assay. And so to do this, we'll essentially repeat our CAR T killing protocol with one slight modification, and that is we will add a therapeutic antibody, rituximab, which will bind to CD20 expressed on our target cells, and our natural killer cells can bind to that antibody and specifically kill our CD20 expressing cells. But we expect these not to harm our cells that lack that CD20 receptor. In this case, this is a very potent killing mechanism, and so we're only going to incubate for 2 hours before analyzing. And so much as we saw previously, it's easy to identify our 3 different cell types and then to determine the percent of our target and our bystander cells that were killed. And so functionally, this 4-color panel is showing us that we can easily identify our different cell types and that we can characterize the killing of one cell type by another. And so functionally, everything looks good here. However, we thought more about this experiment and realized that the natural killer cells we're using are fresh from a human, just a few hours prior to the assay, while the target and bystander cells in this experiment are from a totally different human. And we're also lacking a lot of components here. These cells are not in a human, they're in media, and we're missing a lot of cellular components like serum, like all of the other immune cell types that may contribute either via cytokine signaling or direct signaling to encourage these cells to kill. And so we thought, let's see if we can do the same experiment, but let's do it in whole blood. Let's not isolate out all of the different cell fractions, let's keep them intact. And so let me show you how we accomplish this. First, some background on how this is even possible. Because in whole blood, around less than 1% or even 0.1% of the cells in our blood are leukocytes. The rest are erythrocytes. And those erythrocytes, those red blood cells, can obscure our analysis. And indeed, if we run whole blood through a capillary and take a picture of it, even after some tenfold dilution, we see just this massive amount of cells in a capillary. Using a technique called acoustic focusing, we can use sound waves, ultrasonic sound waves, which actually exert a force on the cells and gently push them into a single file line. And so we can see that the focusing works, but this really doesn't benefit us when we're trying to do single cell analysis. What we need to do is further dilute our sample tenfold and then use acoustic focusing. And now even in whole blood, we can analyze one cell at a time as they pass through our interrogation point in a flow cytometer. Unfortunately, we have a flow cytometer available with this acoustic technology built in called the Attune Nxt. And as you see on the right, this instrument is 10 times faster in terms of volume of sample per minute than other flow cytometers that are available. At the same time, you can analyze up to 14 colors on a 4-laser instrument. And so also for high throughput experiments, this year we launched the Sidekick Max auto sampler, which will allow the analysis of a 96 well plate in only 22 minutes, or a 384 well plate in 88 minutes. So you see the extra fluidics bottles on the right here that help to enable this really dramatic increase in throughput for the system. Now, of course, this is compatible with our own lab automation suite, including robotics and incubation and a fluidics cart, or it can integrate with other versions of lab automation as needed. Now, anyway, if we were to analyze a sample of whole blood on the Attune and dilute that whole blood about 100-fold into media and analyze them, we can analyze single cells in whole blood. The trouble is we now have so many erythrocytes that we can't even visualize our lymphs, monos, and grands. Now, fortunately, the Attune has a feature by which we can use a special side scatter kit that we can detect side scatter on both the violet laser and on the blue laser. And what we're doing here is taking advantage of the fact that our red blood cells absorb light in the violet range of the spectrum, around 405 nanometers. And so without any label required, just based on the intrinsic absorbance of light by our erythrocytes, we can see two populations of cells here. One being this very large population of red blood cells, and then a smaller population above of white blood cells. And so if we can prove that these are white blood cells by labeling whole blood with a dye that binds DNA in live cells. And so by using dicycle violet, we can see a positive population here that is less than 1%, that represents all cells with a nucleus. Our erythrocytes lack a nucleus, and so we see they're dim on this histogram. So if we gate on the positive cells, we can prove to ourselves that, in fact, these are leukocytes that we're visualizing. Now, with flow cytometry, we have the ability to set a fluorescence threshold and teach our instrument to essentially ignore cells, in this case, that lack a nucleus. And so we can now analyze whole blood by flow cytometry, but only capture events if they contain a nucleus. And so we can build on this and now attempt to incorporate our immune cell killing assay in whole blood. And so here's a brief protocol for how we accomplish this. We published this about a year ago, but the brief version is, let's take our cancer cell line, in this case Ramos cells, and we'll label them with cell trace deep red. Then we'll take 100,000 of those cells at 10 to the 7th per mil and spike them into only 5 microliters of whole blood and a final concentration of 10 nanomolar rituximab for a total volume of 25 microliters per well, and as you see here on the right. If we now allow this to incubate for 2 hours, we will then add a very substantial amount of media, 2 mils of media per well. This is possible because we're using a deep well dish where each well holds 2 milliliters of volume. And so essentially we're diluting our whole blood about 500-fold by the time it's analyzed. Now we'll add a viability dye, cytotoxic green, as we've discussed previously, and dicycle violet, which will allow us to threshold only on nucleated cells. Then we'll take this plate, put it into our Attune Nxt autosampler, and analyze every well at 500 microliters per minute, which is about, depending on the instrument, 5 to 10 times faster than other flow cytometers are capable of analyzing. And so we can get through this entire plate actually very quickly. And so what do we see as a result of this? Well, first, we're able to effectively identify only nucleated cells and again, ignore our erythrocytes. And then, because we pre-labeled our cancer cells with a tracking dye, we can easily and clearly resolve our target cells from the white blood cells that are native in that cell population. And so then the next question we wanted to ask is, how viable are the target cells? Were they killed during this 2 hours of sitting in whole blood? And the answer to that is yes, they were very substantially killed. We see 54% of dead cells as reported by cytotoxic green. And so again, we can clearly identify our target and our native white blood cells, and we can very clearly see our live and our dead cell populations. And if you like, we can combine this into a single dual parameter plot, where we can see this very large population of dead target cells. It's also important to note that our white blood cells are not dead. They're very live and healthy after 2 hours of incubation in this experiment. And so our next question was, were these cells killed as a result of the therapeutic antibody that we added, encouraging the killing by both natural killer cells and by complement proteins in our whole blood? And the answer to that is yes. If we leave out that therapeutic antibody, the rituximab, as shown on the bottom, our dead cell population essentially vanishes. So it's only with an appropriate dose of rituximab that we see that killing. And indeed, that entire 96-well plate was a dose-response experiment to characterize what concentration of this antibody is required to encourage, in this case, probably an innate immune response by the components of that whole blood, most likely driven by complement proteins, natural killer cells, and potentially some neutrophils as well. So this type of model where we're not just characterizing the killing of cells, but we're trying to make it as physiologically relevant as possible. We're hoping this will help to better predict the functioning in vivo of therapeutics. So a lot more work is being done on this kind of model system to study killing in whole blood. But we also realize that approximately 10% of all cancers are blood cancers, or liquid tumors, you could call them. The rest are solid cancers, solid tumors. And so for this type of model, we need a different model system. And often fluorescence microscopy is used for that. So we can use a similar setup to study, in this case, the killing of breast cancer cells by natural killer cells. Again, we've labeled our human primary natural killer cells with a tracking dye, so we can see them as blue. When we add these to our SKBR3 cells in the presence of Herceptin, we can see that our breast cancer cells turn from green to a very bright red as they're killed. And this is using a system called the Live Dead Cell Imaging Kit with two components, a green dye that labels our live cells, and then as those cells die, the green signal is lost and a very bright red signal appears. And so functionally, we can see that our NK cells are able to act in an antibody-mediated manner to kill breast cancer cells, to target and kill a solid tumor model. But again, we'd like to make this a more physiologically relevant model. And so we can pivot from 2 dimensions into a 3-dimensional model simply by growing our cells in a special kind of dish called Nucleon Sphera microplates, which in this case we've added 5,000 cells per well and incubated overnight, and we see the presence of 96 uniform spheroids. And we can now add our natural killer cells to each of these spheroids, either in the absence of opsonizing antibody or in the presence of cetuximab, an anti-EGF receptor antibody. And we can see in the left panel, in green, we have a few dead cells. The middle panel, when we add our natural killer cells, we can see that they have penetrated into this tumor spheroid, but there's not much killing that has happened. In the panel on the right, if we add our cetuximab, our specific antibody, then we see a dramatic increase in our green signal. And we can actually quantify this and not only qualitatively, but quantitatively study the killing of solid tumors by natural killer cells. So we've looked at a range of different anti-tumor or anti-cancer immunity mechanisms and a range of different biologies. But I'd like to pivot now, because while T cells and NK cells are very effective at targeting cancer cells, they also are professionally cells that professionally target virus-infected cells, and so not in a drug-mediated fashion, but just by the biology of the cells, they're able to target cells in which viruses have infected. And so we'll look briefly at some different mechanisms, different methods we can use to study this process of viral infection. First, we can look briefly at how viruses are able to enter cells and begin the replication process. Now, different viruses bind and enter cells through different routes. Sometimes this can be through direct fusion with the host cell membrane, or through binding to a cell surface receptor followed by endocytosis. And various viruses will be released from this endocytic pathway at different stages. So either in the early endosome or late endosome or lysosome, depending on the specific biology of a given virus. Now, it's important to note that this pathway is an acidic pathway. So viruses outside the cell are in a neutral pH environment. As they enter an endosome, they become acidified, and as they progress to a late endosome or a lysosome, that pH can drop down to around 4. And so we can take advantage of that to study the uptake and endocytosis and maturation of viral particles. And so in one example, if we take enterovirus and label it with a pH-sensitive dye called Frodo-IFL green, the STP ester is an amine reactive version of this dye. If we label a virus with this dye, it will be non-fluorescent outside a cell, but it will become very brightly fluorescent as it's endocytosed and as it progresses along that endocytic pathway. And so the fluorescent signal by the dye gives us a quantitative measure of how far into a cell that virus has progressed. And in this case, a drug treatment is being used to inhibit acidification of that pathway. So we can see some panels where the signal is less bright. Now, we can perform a quantitative analysis with the same type of pH-sensitive dye, in this case using reovirus, labeled with the red version. So proto-IFL red STP ester was used to label this virus. Then it was added to HeLa cells for the indicated times in this chart. And then with flow cytometry, we can just ask how bright is each cell in that red channel. And as we see in our photo-labeled and treated channel, our mean fluorescence intensity increases quite substantially, 3 to 4-fold brighter. While if ammonium chloride is used to alkalinize those compartments, our signal remains minimal. And so using this approach, we can effectively track the internalization of viruses into cells. So after internalization, we'd like to study the encoding and replication of viruses in host cells. Now, there has been a lot of mention in both the media and in scientific literature this year of a compound called remdesivir that is actually a prodrug. When this is metabolized in humans or in cells, it becomes an active metabolite, as shown here in the middle. This is not too different from the nucleoside adenosine, one of the four bases that is contained in incorporated into replicating DNA. There are, I guess I should call out that this active metabolite functions by once 3 or 4 copies of it are incorporated into an elongating RNA template or a transcript, it will interrupt that process and effectively block the formation of that nascent transcript. We can make additional nucleoside modifications. We can modify uridine with a handle, as shown on the very top right, an ethinyl handle, and create 5-ethinyl uridine. Now, in this case, this very small tag will be incorporated into actively produced RNA. However, it will not inhibit the ability of that RNA to be produced. What it does is gives us a very small tag that if we are to feed this to our cells, this will be incorporated into nascent RNA. Then we can use a click reaction to click on an azide dye conjugate, in this case using an alexafluor dye. And so after incubating our cells with this compound, we can fix and perm and perform a click reaction. And then in the image on the right, we can visualize locations of active RNA production. And so as shown by the bright magenta, we can actually visualize where, in this case, RNA is produced by our mammalian cells. This can become valuable for studying the transcription of viral RNA if we treat our host cells with the compound actinomycin D, which inhibits the cellular DNA-dependent RNA transcription. And so in this case, we're only visualizing synthesis of coronavirus RNA in host cells by the RNA-dependent RNA polymerase. So we can see over time that this coronavirus is very rapidly producing its own RNA, and we see the nucleus is dark because we have blocked the ability of the host cell to transcribe its own RNA. We can transition this into a multiplex experiment combined with antibodies against the coronavirus non-structural protein 12 and an antibody against double-stranded RNA. And as you see in the bottom insets, we can begin to co-localize foci of viral RNA production, the viral protein, and double-stranded RNA in the same place. And so while this is reporting specifically the production of virus RNA, it's not actually reporting any specific template, the RNA that encodes any specific protein. So we can do that, but using a different method. For imaging, we would call this ViewRNA Cell Plus. And in a Zika virus model, we can use an antibody against tubulin and use our ViewRNA Cell Plus to visualize these on the bottom right, bright spots indicating Zika virus RNA localization. Now, for imaging, this is called ViewRNA Cell Plus. For flow, we call this the prime flow RNA assay. And this is compatible with both immunophenotyping and a range of other functional dyes, such as viability. So, for example, using an HIV model, we can use a standard gating scheme to identify our live helper T cells, as shown in this sequence. And then in those live helper T cells, we can ask how much HIV-1 RNA is present in each of those, a variety of different samples. And we can see in the bottom right of each of these density plots, we can identify a very small subset of our helper T cells that actually contain HIV-1 RNA. So now we're detecting a specific transcript unique to a given virus that's contained within a host cell. So not the total presence of the virus, but the presence of an individual RNA transcript. Now with prime flow, there are thousands of different targets available off the shelf, and one of the newest is to specifically detect the SARS-CoV-2 RNA in infected cells. So we recently released a probe set specific to this that covers multiple regions of the SARS-CoV-2 transcript. So there's a link here at the bottom that will direct you to that. But we find this is a very powerful method to characterize the interaction of viruses with host cells. So this and many of the other things that I described today are available through the Immunology at Work Resource Center. The protocols I mentioned today for both isolating, expanding, and characterizing the function of these immune cells are available here. Some other great features, one that I personally use quite often on the right is the Immune Cell Guide, which breaks down all of the different immune cell subsets and what CD markers they express either in a resting or in an activated state. That becomes quite helpful for building panels. There's a QR code at the bottom. If you scan it with your phone, it will take you directly to that website. And so I'd encourage you to take advantage of this resource. We will keep this up to date as the field continues to evolve, but it's meant as a resource for you to continue learning in this area. So I realize these are busy times as we're approaching the end of the year, so I want to thank you for spending this hour with us to focus on both anti-cancer immunity and on antiviral immunity. So thank you for joining us, and I think now we have plenty of time left to take any questions you have. So thank you.
Thank you, Chris, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So our first question is, do you have NK cell expansion from mouse?
Yes, so the thanks for the question. The dynamides are also available for mouse models as well. So either T or NK or monocytes, there's a variety of different ones available for human or mouse. And they're a research grade and also a CTS, meaning a cell therapy approved grade of those beads.
Okay, thank you, Chris. Our next question, how do you activate these human NK cells?
Yeah, so NK cells we can activate with just IL-2 or we can more specifically activate them, to make them think that they're targeting different cell types or viral infected cells with a variety of other cytokines. So those protocols have been built into the Immunology at Work Resource Center. And so really there's a lot of detail there on what cytokine might be appropriate for what model, depending on how you're planning to use your NK cells. So thanks for the question.
Our next question. What culture media system do you use to culture your NK cells?
Good question also, because that's one that there is less available in the literature about what is a good media for NK cells. Of course, there's the optimizer media that's very widely used for T cells. For NK cells, again, there is a media recipe called out specifically in one of the protocols in that Immunology Resource Center that's based on just RPMI, or you can use AIM5 as well. It's a bit of a legacy system. We are working on a much better system, and so again, hopefully we'll see some innovation in that space soon. But for now, the recipe that we used is fully available in protocol in that resource center.
Great, thank you, Chris. Okay, our next question. Do target cells leak cell tracing dye after killing?
Yeah, so that's an insightful question. The cell trace dyes, they label proteins in the target cells. And so they're very stable, very well retained over time. And actually, CFSC used to be used for this, but we found that would leak out quite rapidly over time. The newer colors of cell trace dyes are much more well retained, so you get stable fluorescent signal over time. So immediately after killing, the signal should be unchanged. However, if your cells that are being killed actually start to bleb and fall apart, those blebs will still be fluorescent. You can still see those by flow cytometry. So essentially what you'll see is your target cell will begin to shrink or become less bright, but you can still identify it.
Okay, great. Our next question. Do these dyes affect cell growth?
Oh, yeah. So great question also. So that was something during the testing of these. And again, we'll refer back to CFSE, which is a legacy dye that's now 26 years old, but widely used. But that does affect the ability of cells to grow. It can be toxic. So during the development of the cell trace dyes, meaning the violet, far red, and yellow, we screen those to see if they are toxic. And so we launched those on the basis that they're not toxic to cells. So they're not going to affect the ability of your cells to proliferate. If you're using some really sensitive cell type, I would recommend using a lower concentration of those cells. So, those dyes, 1 micromolar or less to really be sure that it's not having an impact. So, yeah, thanks. Another very good question.
Our next question. Any tools for human gamma delta T cells, isolation or expansion?
Yes, definitely. I think as far as isolation of those, sometimes this just comes down to sorting with FACS, antibody-based immunophenotyping followed by sorting. If there's a specific combination of antibodies that you can identify, it might be possible to cobble together your own bead-based isolation. But generally for these, smaller subsets of cells, we've called out in pretty good detail, actually, in the Immunology at Work Resource Center, how to identify things like gamma deltas, what markers are appropriate, and if you are going to sort these with FACS, what some considerations are for both isolation and growth of those cells.
All right, thank you so much. We have another question here. Can the in-blood ADCC be run on a more basic cytometer that does not have the 405 nanometer SSC?
Yeah, that's a good point. So the whole blood immune cell killing assay, we showed it on the Attune. There are other instruments that are able to look at side scatter with 405, and that's just one way to do it. You can also use just dicycled violet or another live cell DNA dye to label nucleated cells, and then you can just gate on that population and use that approach to ignore erythrocytes. The trouble is you need throughput. Because red blood cells are such a large fraction of your whole blood, you really need the throughput of the Attune to make this a practical assay so that you can actually analyze enough cells in a reasonable amount of time, especially if you're doing a dose response curve or something like I showed. So yes, it's definitely possible on other instruments. It's just not, it's much more practical if you have access to an Attune.
Awesome. Okay, next question. Can we use CD28 dynabeads to check the activation of TIL?
To check the activation of TIL. So meaning tumor-infiltrating lymphocytes that are potentially isolated from a human tumor. The dynabeads, the first example that I showed for isolating T cells, yeah, those would be great for pulling TILs out of a dissociated human tumor, trying to isolate TILs. In fact, we have one that is CD3, CD28, and CD137, so you can start to more accurately isolate activated T cells, or to better activate them. So I would suggest that the dynabeads could be good for pulling T cells out, specifically tumor-infiltrating lymphocytes, out of a lysed tumor, but not so much to detect activated cells. For that, we'd probably use a variety of antibodies against things like CD5, CD69, or potentially the expression of cytokines in those cells. So, yeah, thank you.
Okay, another question. Can cell trace dye be used with calcine?
Sure, yeah, and that combination where calcine is usually referring to calcine green, and it's a dye that I showed during the NK example. That was the dye that turned those live cells green. You could envision a good assay combination where you label, one population of cells with a cell trace dye, and then you interrogate killing, as the perforin and granzyme in your T cells or your natural killer cells lyse the target cells, the calcine will leak out. And so there are some considerations there. Some cancer cells incorrectly turn on a membrane pump that pumps calcine out. And so you need to check and see if that exists in your model. It also exists in stem cells. And so there are some things to be aware of with calcine, but it can become a valuable part of an assay when combined with cell trace dyes.
We have another question. Looks like we have time for just a few more. Do you have any dynabeads to isolate platelets?
That's a good question. It's not my area of expertise, and so I'd have to look and get back to you on that. We do have the ability with the Attune, where I showed you the erythrocyte population and the leukocyte population. In that density plot, you can actually see platelets there as well. So you're able now to directly analyze platelets. They might be labeled with CD41 or ability markers, but you don't so much need to pull them down anymore. You can directly analyze them on that instrument.
Great. We've got one more question. Can prime flow be used to detect only specific RNA transcripts or any target I want?
Oh, yeah. So the prime flow method to detect specific transcripts in cells, we have a wide range of existing targets that are available. But if you have something specific, like you're looking to study the RNA synthesis of something that you've built, like a chimeric antigen receptor in your cells, we can custom make a probe set for whatever you need. So there's a resource on thermofisher.com where you can either search everything that's available off the shelf, or if you need something custom made, we can certainly do that for you and recommend the best route to make that successful for you. So thank you.
Thank you, Chris. Do you have any final comments for our audience?
Sure. Yeah, I want to thank everyone for joining us today during these busy times, and do want to mention two things. One, I encourage you to follow up and visit the Immunology at Work Resource Center and take advantage of, most of the questions that were asked today have an answer somewhere in there, whether it's a protocol or a media type, and also to call out that in 2 weeks, on November 17th, we have another webinar coming up with Sarah DiBiase talking about SARS-CoV-2 infection and how that's studied in cells, specifically using the Attune Nxt Cytometer. So I'll plan to see everyone there in 2 weeks, and we'll see, enjoy Sarah's presentation. So thank you everyone for joining us today.
Thank you again, Chris, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.